Your body and your brain don't necessarily age at the same rate. That's not a metaphor. It's an increasingly well-documented finding in neuroscience research, and it's a genuinely important distinction because a lot of the biological age conversation we've had on this channel, epigenetic clocks, functional physical tests, focuses primarily on the body, cardiovascular fitness, muscle mass, metabolic health. Your brain has its own partially independent aging trajectory, and research increasingly suggests that someone can be in genuinely excellent physical shape while their brain is aging at a considerably faster rate, or vice versa. I want to walk you through how researchers actually measure brain age, specifically what drives divergence between brain age and body age, what the early warning signs actually look like, and what the evidence says about slowing or even partially reversing this specific trajectory. Let's start with how brain age gets measured, because I think this deserves genuine, specific explanation before we get into anything else. The most established research method involves structural MRI, magnetic resonance imaging of the brain, analyzed using machine learning algorithms trained on large data sets of brain scans from people of known chronological age. These algorithms learn to identify patterns of brain structure, gray matter, volume, white matter integrity, and the size of the ventricles, the fluid-filled spaces within the brain that tend to enlarge as surrounding brain tissue shrinks with age that reliably correlate with chronological age. And once trained, the algorithm can be applied to a new person's brain scan to estimate their brain age, the age their brain structurally resembles, regardless of their actual chronological age. Citation: this structural MRI-based brain age estimation methodology has been established extensively in neuroimaging research, published in journals including Neuroimage and Nature Communications, with foundational work by researchers including James Cole, developing widely used brain age prediction models. The number that actually matters here is what researchers call the brain age gap, the difference between someone's predicted brain age and their actual chronological age. A positive gap, meaning the brain appears older than the person's actual age, has been consistently associated in research with elevated risk for cognitive decline, dementia, and, in some studies, elevated all-cause mortality risk. Citation: this relationship between a positive brain age gap and elevated risk for cognitive decline, dementia, and mortality has been examined extensively in neuroimaging and neuroscience research published in journals including Molecular Psychiatry and Nature Communications. I want you to notice something about the language here. This isn't simply describing normal aging happening on schedule. It's specifically describing acceleration, a brain that looks structurally older than the calendar would predict, which is precisely the kind of signal that, similar to the epigenetic clocks we discussed in a previous video, appears to carry genuine, independent predictive information beyond chronological age alone. Let me address what's actually happening structurally when a brain shows this kind of accelerated aging pattern, because I think the specific anatomical changes matter for understanding this whole conversation. Gray matter volume, the brain tissue containing neuronal cell bodies where most information processing actually happens, tends to decline with age, but the rate and specific pattern of this decline varies considerably between individuals, and research has found that certain regions, particularly the prefrontal cortex, involved in executive function and decision-making, and the hippocampus, centrally involved in memory formation, tend to show earlier and more pronounced volume loss in individuals with accelerated brain aging, specifically citation. This pattern of region-specific gray matter volume loss associated with accelerated brain aging has been examined in neuroimaging research published in journals including Neurobiology of Aging and Brain. White matter, the tissue containing the long, myelin-insulated nerve fibers connecting different brain regions to each other, also shows age-related changes, with research finding that declining white matter integrity, sometimes measured through a technique called diffusion tensor imaging that tracks how water molecules move along these nerve fiber pathways, is associated with slower processing speed and reduced cognitive flexibility in individuals showing accelerated patterns, citation. This relationship between white matter integrity decline and cognitive processing changes has been examined extensively in diffusion tensor imaging research published in journals including Neurobiology of Aging. I want to bring in something about why brain age and body age can diverge from each other in the first place. Because I think this is genuinely one of the more important, practically useful pieces of this entire conversation. Research examining the relationship between physical biological age markers, like the epigenetic clocks we discussed previously, and brain age specifically has found meaningful but genuinely imperfect correlation between the two, meaning they're related but distinguishable measurements, each capturing something the other doesn't fully account for. Citation: this imperfect correlation between systemic biological aging markers and brain-specific aging measurements has been examined in geroscience research, published in journals including Nature Aging. This makes considerable biological sense once you think through the mechanisms involved. Your brain sits behind the blood-brain barrier, a specialized, selectively permeable structure that regulates what substances can pass from your bloodstream into brain tissue, meaning your brain's exposure to circulating inflammatory markers, metabolic byproducts, and other factors influencing aging elsewhere in your body is genuinely partially buffered compared to more directly exposed peripheral tissues. Citation: this role of the blood-brain barrier in partially buffering the brain from systemic circulating factors relevant to peripheral tissue aging has been discussed in neurovascular biology research published in journals including Nature Reviews Neuroscience. At the same time, your brain has genuinely unique vulnerabilities of its own extraordinarily high metabolic demand relative to its size, meaning it's particularly sensitive to disruptions in blood flow and oxygen delivery, and specific susceptibility to the accumulation of misfolded proteins, amyloid and tau specifically, implicated in Alzheimer's disease, that don't have a direct equivalent in most other organ systems we've discussed throughout this channel. Let me talk about cardiovascular health's relationship to brain aging specifically, since I think this represents one of the more genuinely well-established, actionable connections in this entire body of research, and it directly ties this conversation back to content we've covered extensively elsewhere on this channel. Research has consistently found that cardiovascular risk factors, hypertension, elevated cholesterol, diabetes, and reduced cardiorespiratory fitness specifically, are associated with accelerated brain aging and increased dementia risk, and this relationship makes considerable mechanistic sense given how metabolically demanding brain tissue is and how directly it depends on efficient, healthy blood flow. Citation: this relationship between cardiovascular risk factors and accelerated brain aging and dementia risk has been examined extensively in research published in journals including The Lancet Neurology and Circulation. I want to bring in something specific here connecting directly to our previous video on VO2 max, since I think it deserves genuine emphasis given that video's focus. Research has specifically found that higher cardiorespiratory fitness is associated with larger hippocampal volume, and in intervention research, that improving cardiorespiratory fitness through structured aerobic training can produce measurable increases in hippocampal volume, even in previously sedentary older adults, a finding we touched on briefly in that previous video that deserves fuller emphasis here given this video's specific focus on brain aging. This is genuinely one of the more concrete, actionable connections in this entire conversation. The same cardiorespiratory training we've discussed extensively for its cardiovascular and longevity benefits appears to directly, measurably influence brain structure and brain aging trajectory specifically. Let me address something about midlife hypertension specifically, since I think it deserves particular emphasis given how consistently it's emerged in this research. Longitudinal research following individuals from midlife into older age has found that hypertension, specifically measured during midlife, rather than hypertension that develops later in life, shows a particularly strong association with subsequent dementia risk and accelerated brain aging, suggesting that the cumulative vascular damage from years of elevated blood pressure during this specific life stage may be particularly consequential for long-term brain health. Citation: this specific association between midlife hypertension and subsequent dementia and brain aging risk has been examined in landmark longitudinal research published in journals including The Lancet and JAMA Neurology, including findings from long-running cohort studies specifically designed to track cardiovascular risk factors from midlife through older age. This offers genuinely important practical context. If you're in your 40s or 50s and have been putting off addressing borderline elevated blood pressure, this research suggests the stakes for your long-term brain health specifically, not just your cardiovascular system generally, may be higher than commonly appreciated during this specific window of life. I want to address sleep's relationship to brain aging in considerable depth now, because I think this represents one of the more mechanistically fascinating and genuinely actionable pieces of this entire conversation. Research has identified a specific physiological process called the glymphatic system, a network that becomes considerably more active during sleep, specifically functioning to clear metabolic waste products, including amyloid beta, the protein that aggregates to form the plaques characteristic of Alzheimer's disease, from brain tissue. Citation: this glymphatic system and its role in clearing metabolic waste, including amyloid beta, from brain tissue specifically during sleep, was established through foundational research by Nedergaard and colleagues, published in Science, and has become a central framework for understanding sleep's role in brain health. This offers a genuinely direct, mechanistic explanation for why chronic sleep insufficiency has been so consistently associated with elevated dementia risk in longitudinal research. Inadequate sleep appears to directly compromise your brain's capacity to clear exactly the kind of protein buildup implicated in neurodegenerative disease. Let me bring in the specific longitudinal research connecting sleep duration to subsequent dementia risk here, since I think the actual data deserves direct citation rather than remaining at a purely mechanistic level. Research following large cohorts over extended periods has found that both consistently short sleep duration, generally defined as 6 hours or less nightly, and in some research, consistently long sleep duration, have been associated with elevated subsequent dementia risk, with one particularly notable study finding that persistent short sleep duration, specifically during midlife, around age 50 and 60, was associated with meaningfully elevated dementia risk assessed decades later. Citation: Sabia and colleagues. This research finding an association between midlife short sleep duration and subsequently elevated dementia risk assessed decades later was published in Nature Communications. I want to bring in something about sleep architecture specifically here as well, beyond simply total duration, since research has found that reduced deep, slow-wave sleep specifically, the sleep stage most associated with the glymphatic clearance process we just discussed, shows a particularly strong association with subsequent cognitive decline and amyloid accumulation, independent of total sleep duration alone. Citation: this relationship between reduced slow-wave sleep specifically, independent of total sleep duration, and subsequent cognitive decline and amyloid accumulation has been examined in sleep and neurodegeneration research published in journals including Science Translational Medicine. Let me address hearing loss here as well, since this represents a genuinely underappreciated but increasingly well-established risk factor for accelerated brain aging that I think deserves direct, dedicated attention. Research has found that hearing loss, particularly moderate to severe hearing loss left uncorrected, is associated with meaningfully elevated risk for cognitive decline and dementia, and a large influential analysis specifically identified hearing loss as one of the largest single modifiable risk factors for dementia among a broader list of identified modifiable factors. Citation: Livingston and colleagues. This identification of hearing loss as a major modifiable dementia risk factor was part of a comprehensive analysis published in the Lancet Commission report on dementia prevention, intervention, and care. The proposed mechanisms here are genuinely interesting and worth understanding. Hearing loss appears to contribute to cognitive decline through several distinct pathways: increased cognitive load, your brain having to work considerably harder to process degraded auditory signals, leaving fewer cognitive resources available for other tasks, reduced sensory input to the brain more broadly, potentially contributing to accelerated atrophy in auditory processing regions and their connected networks, and, genuinely importantly, social isolation, since untreated hearing loss frequently leads people to withdraw from conversation and social engagement, removing exactly the kind of cognitively stimulating social interaction that appears protective against cognitive decline. Citation: these multiple proposed mechanisms connecting hearing loss to cognitive decline, including increased cognitive load, reduced sensory input, and secondary social isolation, have been discussed extensively in research published in JAMA Neurology and related audiology and neurology literature. This is genuinely actionable information. If you or someone you know has noticed hearing difficulty and has been putting off addressing it, this research suggests hearing aids or other appropriate correction may carry meaningful benefit for brain health specifically, well beyond simply making conversation easier. Let me talk about depression and its relationship to accelerated brain aging here as well, since I think this deserves careful, genuine treatment given how common depression is and how directly it connects to themes we've covered in previous videos on this channel. Research has found associations between depression, particularly recurrent or chronic depression, and accelerated brain age, along with reduced hippocampal volume specifically. Citation: this relationship between depression and accelerated brain aging, including reduced hippocampal volume, has been examined in psychiatric neuroimaging research published in journals including molecular psychiatry and biological psychiatry. I want to be genuinely careful here about the direction of causality, since this relationship almost certainly runs in multiple directions simultaneously. Depression may contribute to accelerated brain aging through mechanisms including chronic stress and elevated cortisol, which we discussed in a previous video as having genuine documented effects on brain structure, while pre-existing changes in brain structure and function may themselves contribute to the development of depressive symptoms in the first place, meaning this represents a genuinely complex bidirectional relationship rather than a simple one-directional cause and effect. I don't want anything in this video to suggest that experiencing depression means your brain is definitely irreversibly aging faster. This represents an association observed at the population level in research, not a certain individual prognosis, and proper treatment for depression, which we discussed in our previous video on cognitive behavioral therapy and self-talk, remains genuinely important both for its own sake and, this research suggests, potentially for long-term brain health as well. Let me bring in cognitive reserve here as a genuinely important concept, since I think it adds crucial nuance to how you should interpret everything we've discussed throughout this video so far. Cognitive reserve refers to the brain's capacity to maintain normal cognitive function despite the presence of underlying brain pathology or age-related structural changes, essentially reflecting the idea that some brains can tolerate and compensate for more accumulated structural damage before that damage translates into noticeable functional cognitive decline. Citation: this cognitive reserve concept and its role in explaining why some individuals maintain cognitive function despite significant underlying brain pathology was established through foundational research by Yakov Stern and colleagues, published extensively in journals including The Lancet Neurology, and subsequently elaborated across a substantial body of subsequent research. Research examining what actually builds cognitive reserve has found associations with educational attainment, occupational complexity, meaning jobs involving greater cognitive demand and complexity, ongoing engagement in cognitively stimulating leisure activities throughout life, and connecting directly to the social connection theme we discussed in a previous video, rich social engagement. Citation: these factors associated with building and maintaining cognitive reserve have been examined extensively in research on cognitive reserve and dementia risk published in the Lancet Neurology and related neuroepidemiology literature. This is genuinely important, hopeful context. It suggests that the relationship between brain structural aging and actual functional cognitive decline isn't perfectly linear or deterministic. Someone with genuinely accelerated brain age on structural imaging may nonetheless maintain considerably better functional cognitive performance than that structural finding alone would predict if they've built meaningful cognitive reserve through education, engagement, and social connection throughout their life. I want to spend real time now on what actually predicts and drives the development of dementia specifically, since I think grounding this entire conversation in the most comprehensive current research on modifiable risk factors adds genuine practical value here. The Lancet Commission on Dementia Prevention, a large, influential group of researchers who periodically synthesize the entire body of evidence on modifiable dementia risk factors, has identified a specific set of factors estimated to collectively account for a substantial portion, roughly 40% by their most recent analysis of dementia cases worldwide, at least theoretically preventable or delayable through addressing these specific modifiable factors. Citation: this comprehensive Lancet Commission analysis identifying modifiable dementia risk factors and their estimated collective contribution to dementia risk was published in the Lancet, with periodic updates incorporating newer research as it becomes available. These factors span essentially every life stage: in early life, less education, in midlife, hearing loss, which we discussed in detail, traumatic brain injury, hypertension, excessive alcohol consumption, obesity, and in more recent updates to this analysis, air pollution exposure and untreated vision loss, and in later life, smoking, depression, social isolation, physical inactivity, diabetes, and notably hearing loss appearing again as one of the single largest individual contributors across this entire framework. Citation: this comprehensive list of modifiable risk factors spanning early, mid, and late life has been detailed in the full Lancet Commission report on dementia prevention, intervention, and care. Let me bring in something about traumatic brain injury specifically here, since it's a factor I mentioned but haven't yet explained in genuine detail, and I think it deserves fuller treatment given how relevant it is to a broad range of common activities. Research has found that even a single moderate traumatic brain injury and, in some research, repeated milder head impacts of the kind that can occur in certain contact sports or activities are associated with meaningfully elevated subsequent dementia risk, with the association generally appearing stronger with greater injury severity and greater cumulative exposure to head impacts over time. Citation: this relationship between traumatic brain injury history, including repeated milder impacts, and elevated subsequent dementia risk has been examined extensively in neurology research published in journals including JAMA Neurology and the Lancet Neurology. This is genuinely relevant, practical information for anyone involved in activities carrying meaningful head injury risk, appropriate protective equipment, proper technique, and genuinely respecting concussion protocols rather than pushing through symptoms represents a genuinely evidence-supported way of protecting long-term brain health, distinct from but complementary to the cardiovascular and lifestyle factors we've emphasized throughout most of this video. Let me address exercise's relationship to brain health in considerably more mechanistic depth here now, since I think it deserves fuller treatment given how central it's been to our overall channel content, beyond simply the cardiorespiratory fitness and hippocampal volume connection we already discussed. Exercise appears to influence brain health through several genuinely distinct mechanisms operating simultaneously. We discussed brain-derived neurotrophic factor, BDNF, in a previous video regarding exercise and cognitive function, and it deserves reiteration here, specifically in the context of brain aging. Exercise reliably increases circulating BDNF, a protein directly involved in neuroplasticity, the growth of new neural connections, and, in some research, the generation of new neurons specifically within the hippocampus, a process called neurogenesis that continues to a modest but genuine degree throughout adult life. Citation: this role of exercise in stimulating BDNF release and supporting adult hippocampal neurogenesis has been examined extensively in exercise neuroscience research published in journals including Nature Reviews Neuroscience and the Journal of Neuroscience. Beyond the BDNF and neurogenesis pathway, exercise also appears to reduce chronic systemic inflammation, connecting to the inflammaging concept we've discussed extensively across our biological aging content. And chronic neuroinflammation specifically has become increasingly implicated as a genuine contributor to neurodegenerative processes, meaning exercise's broadly anti-inflammatory effects likely contribute directly to brain health through this pathway as well. Distinct from the more direct cardiovascular and BDNF-related mechanisms we've discussed, citation: this relationship between exercise's anti-inflammatory effects and their relevance to neurodegeneration and brain aging specifically has been examined in neuroimmunology research published in journals including Nature Reviews. Neurology. I want to bring in something about the myokine research we covered extensively in a previous video here as well, since it connects directly to this specific conversation, given that we specifically discussed muscle-derived signaling molecules, including irisin, showing evidence of neuroprotective effects, and BDNF contribution directly from exercising muscle tissue itself in that earlier video. meaning the muscle-brain signaling conversation we had previously isn't a separate topic from this brain aging discussion. It represents one of the genuinely important direct mechanistic pathways connecting the two. Let me talk about diet and its relationship to brain aging specifically. Now, connecting to nutrition content we've covered elsewhere on this channel, but focusing here specifically on brain-relevant findings. Research has examined several specific dietary patterns in relation to brain health and dementia risk, with the Mediterranean diet and a specifically brain-focused variant called the MIND diet, combining elements of the Mediterranean diet with additional emphasis on specific foods including leafy greens, berries, and nuts, showing consistent associations with reduced dementia risk and, in some research, slower rates of cognitive decline and brain aging. Specifically, citation, this research on the MIND diet and its association with reduced dementia risk and cognitive decline has been examined in nutritional neuroepidemiology research published in journals including Alzheimer's and dementia. I want to be appropriately calibrated here about the strength of this specific dietary pattern research, since much of it remains observational rather than fully controlled, randomized intervention research, similar to caveats we've applied to dietary pattern research throughout this entire channel. But the general direction of these findings is broadly consistent with the anti-inflammatory, cardiovascular supportive dietary principles we've discussed extensively elsewhere. Let me bring in something about the specific relationship between blood sugar regulation and brain aging here as well, since I think this deserves particular emphasis given how directly it connects to metabolic health content we've covered throughout this channel. Research has found associations between insulin resistance and type 2 diabetes and both accelerated brain aging and elevated dementia risk, with some researchers proposing the term type 3 diabetes specifically to describe the apparent role of impaired brain insulin signaling in Alzheimer's disease pathology. Though I want to be genuinely clear, this remains a more contested, actively debated terminology and mechanistic framework within the field, rather than a fully settled, universally accepted characterization. Citation, this research examining insulin resistance and its relationship to accelerated brain aging and dementia risk, including the more contested type 3 diabetes framework, has been discussed in neuroendocrinology and dementia research, published in journals including the Journal of Alzheimer's Disease and Diabetologia. This offers yet another genuine, direct connection between the metabolic health themes we've covered extensively throughout this channel, muscle's role in glucose disposal, protein and blood sugar regulation, and this specific conversation about brain aging, chronic metabolic dysfunction appears to have genuine, direct consequences for brain health specifically, well beyond the cardiovascular consequences we more commonly discuss. Let me address something about early warning signs now, since I think this represents the genuinely practical piece of this entire conversation that a lot of people watching this video actually want, some sense of what to actually pay attention to in their own daily experience. I want to be genuinely careful and precise here because I don't want anything in this section to encourage inappropriate self-diagnosis or unnecessary anxiety. Normal aging does involve some genuine expected changes in cognitive processing speed and certain aspects of memory, and distinguishing genuinely concerning patterns from normal age-related variation requires proper clinical evaluation, not simply comparing yourself against a list in a video. That said, research on the earliest subtle signs that sometimes precede more obvious cognitive decline has identified some genuinely useful patterns worth being aware of. Difficulty with what researchers call dual tasking, performing two cognitively demanding tasks simultaneously, something we discussed in a previous video in the specific context of dual task walking testing, has been found in research to sometimes represent an earlier, more subtle signal than single task cognitive testing alone would reveal. Citation, this relevance of dual task performance as a potentially earlier, more sensitive marker of subtle cognitive changes has been examined in geriatric neurology research we referenced in our previous video on gait speed and cognition. Changes in gait speed itself, connecting directly to that same previous video, have similarly been found to sometimes precede more obvious cognitive symptoms by a meaningful margin. Difficulty with word finding specifically, more than general forgetfulness, has also been examined as a potentially earlier marker in some research, though I want to be genuinely clear that occasional word finding difficulty is an extremely common normal experience for people of essentially any age, particularly under stress or fatigue. And the research specifically concerns persistent, progressively worsening patterns rather than occasional situational lapses that essentially everyone experiences from time to time. Let me address something about subjective cognitive complaints more directly here as well, since this represents a genuinely interesting, if somewhat complicated, area of research. Research examining individuals who report subjective concerns about their own memory or cognitive function, even when formal cognitive testing shows entirely normal performance, has found that this specific population shows somewhat elevated subsequent risk for progression to more objective cognitive impairment over time compared to individuals without such subjective concerns, though the majority of people reporting subjective cognitive complaints do not go on to develop dementia, meaning this represents an elevated relative risk within a population where the absolute risk and the majority outcome remains reassuring rather than alarming. Citation: this research on subjective cognitive complaints and their relationship to subsequent objective cognitive decline risk has been examined in longitudinal neurology research published in journals including Neurology and Alzheimer's Dementia. I mention this specifically because I don't want anyone watching this video who has occasionally worried about their own memory to walk away unnecessarily alarmed. The research here describes a modest elevation in relative risk within a population where most people experiencing these subjective concerns do not go on to develop significant cognitive impairment, and genuine, persistent concern is best addressed through proper clinical evaluation rather than self-diagnosis based on a general population statistic. Let me talk about formal cognitive testing and brain age assessment accessibility here as well, since I think practical guidance matters given everything we've discussed throughout this video. Formal brain age assessment through the kind of structural MRI research we discussed at the start of this video remains largely confined to research settings and specialized clinical contexts, and isn't something most people have convenient, routine, affordable access to, similar to the epigenetic clock testing accessibility limitations we discussed in a previous video. What is considerably more accessible is proper clinical cognitive screening, which a primary care physician or a specialist in neurology or geriatric medicine can perform using validated, standardized cognitive assessment tools. And I genuinely encourage anyone with persistent, meaningful concern about their own cognitive function or that of a family member to pursue this kind of proper clinical evaluation rather than relying purely on informal self-assessment or general population research statistics like the ones we've discussed throughout this video. Let me bring this entire considerably long conversation together now into what I actually want you to walk away with. Your brain has a genuinely distinct, partially independent aging trajectory from the rest of your body, measurable through structural neuroimaging research that identifies patterns of gray and white matter change correlating with, and in some individuals, meaningfully accelerating beyond chronological age. This divergence between brain age and body age reflects the brain's unique physiological context, partial protection from the blood-brain barrier, alongside genuinely unique vulnerabilities, including extraordinarily high metabolic demand and susceptibility to specific protein accumulation processes not present in most other tissues we've discussed throughout this channel. Critically, and this connects everything we've discussed to a broader, genuinely hopeful theme, a substantial portion of what drives accelerated brain aging appears to be modifiable cardiovascular health, particularly midlife blood pressure management, sleep quality, specifically related to the glymphatic clearance process, treating hearing loss, managing depression, addressing metabolic health, protecting against traumatic brain injury, maintaining regular exercise for its direct effects on BDNF and neurogenesis, and building cognitive reserve through education, cognitively stimulating engagement, and rich social connection throughout life all appear to genuinely, measurably influence this specific trajectory. If there's one thing I want you to take from everything we've covered today, it's this. Brain aging isn't a fixed, purely genetically determined process happening independently of everything else discussed throughout this channel. Sleep, exercise, cardiovascular health, social connection, mental health. It's deeply mechanistically interconnected with all of it, and the same fundamental lifestyle pillars that support the rest of your body's healthy aging trajectory appear to carry direct, substantial and increasingly well-mapped benefit for your brain specifically as well. Let me add considerably more depth here, since there are several important threads worth developing further given how central this topic is to everything else we've covered across this channel. I want to go deeper into the specific machine learning methodology underlying these brain age prediction models, because I think understanding this in more detail helps clarify exactly what these algorithms are and aren't capturing. These models are typically trained using a technique where the algorithm is given thousands of brain scans from healthy individuals of known chronological age and learns to identify the specific structural features, patterns of gray matter thickness across different cortical regions, ventricular volume, white matter hyperintensities, small areas of altered signal on MRI thought to reflect small vessel disease and reduced blood flow that most reliably predict age within this training population. Citation, this machine learning methodology underlying brain age prediction and the specific structural features these models learn to weight most heavily has been described extensively in neuroimaging methodology research published in journals including Neuroimage. One genuinely important methodological consideration here, and one that connects to limitations we've discussed with other biological aging measurements throughout this channel, is that these models are only as good as the training data used to build them, and research has found that brain age prediction accuracy can vary somewhat depending on the demographic composition of the training data set relative to the population being tested, similar to concerns we raised about epigenetic clocks in a previous video. Citation. This consideration regarding training data representativeness and its effect on brain age prediction accuracy has been discussed in neuroimaging methodology literature, examining generalizability of these models across different populations. Let me bring in something about white matter hyperintensities specifically here, since I mentioned them briefly, but I think they deserve fuller, dedicated treatment given how directly they connect to the cardiovascular themes we've emphasized throughout this video. These small areas of altered signal on brain MRI, appearing as bright spots on certain imaging sequences, are believed to reflect small vessel disease, essentially subtle damage to the tiny blood vessels supplying deep brain tissue. And research has found a strong, consistent relationship between the burden of these white matter hyperintensities and both cardiovascular risk factors, particularly hypertension, and subsequent cognitive decline and dementia risk. Citation, this relationship between white matter, hyperintensity, burden, cardiovascular risk factors, and cognitive decline has been examined extensively in neuroimaging and cerebrovascular research published in journals including Stroke and Neurology. This offers genuinely direct, visible evidence connecting the cardiovascular health themes we've discussed throughout this video and throughout this entire channel more broadly to actual visible structural brain changes. These aren't simply statistically associated risk factors. They appear to directly, visibly damage the small vessel network supplying your brain tissue in a way that's genuinely observable on standard clinical imaging. I want to spend more time on the specific relationship between blood pressure variability, not just average blood pressure level, and brain aging here, since I think this represents a genuinely underappreciated, more recently emerging piece of this research. Some research has found that beyond simply average blood pressure readings, greater variability in blood pressure, meaning larger fluctuations between readings over time, is independently associated with elevated dementia risk and accelerated brain aging, potentially reflecting greater cumulative stress on the delicate small vessel network we just discussed. Citation, this research on blood pressure variability, independent of average blood pressure level, and its relationship to cognitive decline and brain aging has been examined in cardiovascular and neurology research published in journals including Hypertension and Neurology. This adds a genuinely useful additional layer of nuance for anyone tracking their own blood pressure, specifically for brain health reasons. Consistency of control may matter in a way that isn't fully captured by simply looking at your average reading or your most recent single measurement alone. Let me address something about the specific relationship between atrial fibrillation and brain aging here as well, since this represents a genuinely important, if somewhat specialized, cardiovascular condition worth mentioning given its prevalence, particularly in older populations. Atrial fibrillation, an irregular heart rhythm, has been associated in research with elevated dementia risk through mechanisms including both an elevated risk of clinically apparent stroke and, importantly, elevated risk of what researchers call silent strokes, small areas of brain tissue damage from reduced blood flow that don't produce obvious, immediately recognizable stroke symptoms, but nonetheless contribute cumulatively to brain tissue damage and accelerated cognitive decline over time. Citation, this relationship between atrial fibrillation, both clinically apparent and silent strokes, and elevated dementia risk has been examined extensively in cardiology and neurology research published in journals including the European Heart Journal and Circulation. This is genuinely important context for anyone with a diagnosis of atrial fibrillation or anyone experiencing symptoms that might suggest it. Proper management of this specific condition carries brain health implications well beyond its more commonly discussed stroke risk in the general sense. I want to bring in something about oral health and its relationship to brain aging here as well, since I think this represents a genuinely surprising, increasingly researched connection that deserves at least dedicated mention. Some research has found associations between chronic periodontal disease, gum disease, and elevated risk for cognitive decline and dementia, with proposed mechanisms including chronic low-grade systemic inflammation triggered by ongoing oral bacterial infection, connecting to the broader inflammaging themes we've discussed throughout this channel, and in some more specific research, the direct detection of certain oral bacteria and their associated toxins within brain tissue itself in post-mortem examination of individuals who had Alzheimer's disease. Citation, this research examining periodontal disease and its association with cognitive decline, including the detection of oral bacterial products in brain tissue, has been published in research, including studies in journals, including Science Advances in the Journal of Alzheimer's Disease, though I want to be genuinely clear that this specific line of research, while genuinely intriguing, remains somewhat earlier stage and more actively debated within the field compared to the more extensively established cardiovascular and sleep-related connections we discussed earlier in this video. I mention this specifically because it's a genuinely underappreciated, low-cost, accessible health behavior, proper dental hygiene, and addressing gum disease that carries potential relevance to this entire conversation well beyond simply oral health in isolation. Let me spend time now on the specific relationship between vision loss and cognitive decline, since I mentioned this briefly earlier, but I think it deserves fuller treatment given its inclusion in the more recent Lancet Commission analysis we discussed. Research has found that uncorrected vision impairment, similar to the mechanisms we discussed for hearing loss, is associated with elevated dementia risk through proposed mechanisms including increased cognitive load from processing degraded visual information, reduced overall sensory input to the brain, and, similarly to hearing loss, an increased likelihood of social withdrawal and reduced engagement in cognitively stimulating activities that depend on adequate vision, reading, for instance. Citation, this relationship between uncorrected vision impairment and elevated dementia risk and its proposed mechanisms has been examined in ophthalmology and neuroepidemiology research that informed its inclusion in the updated Lancet Commission analysis on modifiable dementia risk factors. This is genuinely another example of a common, highly correctable condition, in this case, simply wearing appropriate corrective lenses or addressing treatable eye conditions like cataracts that carries brain health implications that most people wouldn't intuitively connect to this specific topic. I want to address something about the specific relationship between alcohol consumption and brain aging in more mechanistic depth here now, since I think this deserves fuller treatment beyond a passing mention. Research has found that heavy, chronic alcohol consumption is associated with measurable brain volume reduction and accelerated brain aging, with some research finding that even moderate consumption levels, previously considered relatively safe based on older research, may carry some meaningful risk for brain-specific effects, even if the relationship with certain other health outcomes appears more neutral or even mixed at moderate consumption levels. Citation, this research on alcohol consumption, including moderate levels, and its relationship to brain volume and accelerated brain aging has been examined in large-scale neuroimaging research published in journals including Nature Communications and the BMJ. I want to be genuinely careful and precise here, since alcohol and health research more broadly remains an area with genuine, ongoing scientific debate about how to properly interpret observational data, given various confounding factors. But the specific brain imaging research in this area has generally found a more consistently unfavorable pattern for alcohol, specifically related to brain volume and structure, compared to some of the more mixed findings in broader cardiovascular research on moderate alcohol consumption. Let me bring in something about air pollution's specific effects on brain aging here as well, extending on a point I mentioned only briefly in relation to the Lancet Commission analysis. Research has found that chronic exposure to air pollution, particularly fine particulate matter, is associated with accelerated brain aging on structural imaging and elevated dementia risk, with some research finding that particulate matter pollutants may be capable of directly entering brain tissue through the olfactory nerve, essentially bypassing some of the protective function of the blood-brain barrier we discussed earlier in this video, and triggering direct neuroinflammatory responses within brain tissue itself. Citation: this research on air pollution's direct entry into brain tissue via the olfactory pathway and its neuroinflammatory effects has been examined in environmental neuroscience research published in journals including Environmental Health, Perspectives, and the Journal of Alzheimer's Disease. This adds genuinely important additional context to our earlier discussion of the blood-brain barrier as a partial protective mechanism. Certain environmental exposures appear capable of at least partially circumventing that protection through alternative anatomical routes, which is worth being aware of, particularly for anyone living in an area with consistently poor air quality. I want to spend time now on cognitively stimulating activities, specifically since I think this deserves more concrete, specific treatment beyond the general cognitive reserve concept I introduced earlier. Research examining specific types of cognitively engaging activity in relation to dementia risk has generally found that activities involving genuine novelty and challenge, learning a new skill entirely, rather than simply repeating a familiar, well-practiced activity, tend to show stronger associations with cognitive benefit. Connecting to something we discussed in our video on dopamine and novelty, since genuinely novel challenges appear to require and therefore strengthen a broader range of neural circuitry than repeating an already well-mastered task. Citation: this research suggesting genuinely novel, challenging cognitive activities may carry particular benefit for cognitive reserve compared to repeating already familiar activities has been examined in cognitive aging research published in journals including Psychological Science and Neurology. I want to bring in something about the specific research on learning a new language later in life here as well, since it represents one of the more concretely studied examples of this principle, with some research finding that bilingualism, and in some studies actively learning an additional language later in life specifically, is associated with a somewhat later average age of dementia symptom onset, even after accounting for underlying disease pathology, suggesting this specific kind of ongoing cognitive challenge may genuinely contribute to cognitive reserve capacity. Citation: this research on bilingualism and its association with delayed dementia symptom onset has been examined in neurolinguistics and dementia research published in journals including Neurology. Let me address the social connection piece of this in considerably more depth here as well, since I only touched on it briefly earlier, despite how directly it connects to what we've covered throughout this channel, and I think it deserves fuller treatment given the strength of the underlying research. Research examining social isolation and loneliness specifically as distinct concepts, since someone can be objectively socially isolated while not subjectively feeling lonely, and vice versa, has found that both dimensions independently predict elevated dementia risk, with proposed mechanisms including reduced cognitive stimulation from social interaction itself, the chronic stress and inflammatory pathways associated with loneliness specifically that we discussed in a previous video on biological aging, and reduced likelihood of having someone available to notice and flag early cognitive changes that the affected individual themselves might not recognize. Citation: this research distinguishing objective social isolation from subjective loneliness and their independent relationships to dementia risk has been examined in social epidemiology and neurology research published in journals including Lancet, Neurology and Aging, and Mental Health. Let me bring this considerably expanded, thorough conversation to its actual final close now. Everything we've added in this deeper exploration, the specific machine learning methodology and its genuine training data limitations, white matter hyperintensities as visible, direct evidence connecting cardiovascular damage to brain structure, blood pressure variability as an independent factor beyond simple average readings, atrial fibrillation and silent stroke risk, the genuinely surprising oral health and periodontal disease connection, vision loss as another correctable sensory factor alongside hearing loss, alcohol's specific effects on brain volume even at moderate consumption levels, air pollution's direct entry into brain tissue via the olfactory pathway, the particular value of genuinely novel cognitive challenges like language learning for building cognitive reserve, and the distinct independent contributions of objective social isolation and subjective loneliness. All of it deepens and reinforces the central theme this entire video has been built around. Brain aging is not a separate, isolated process happening independently of everything else we discuss on this channel. It is deeply mechanistically woven into your cardiovascular health, your sleep quality, your metabolic function, your sensory health, your social connection, and your ongoing cognitive engagement. And the accumulating evidence increasingly suggests that a substantial, genuinely meaningful portion of your individual brain aging trajectory remains within your influence through the same fundamental lifestyle factors that determine so much of the rest of your long-term health. This video discusses general neuroscience and geroscience research for informational and educational purposes only and does not constitute personalized medical advice or a diagnostic tool. Cognitive changes and concerns about memory or brain health should be evaluated by a qualified physician or neurologist who can provide proper clinical assessment beyond what general educational content like this can offer. If you or someone you know is experiencing persistent, worsening cognitive symptoms, please seek professional medical evaluation rather than relying on self-assessment.